Good morning, everyone. It's 10 o'clock on the dot, so we will get started. Um, just wanted to first give a quick introduction to this workshop and webinar series hosted by the 3Rs Collaborative and IQ Consortium. And the topic of today's workshop is immune competent and lymph node MPS. A quick disclaimer that this webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither the 3Rs Collaborative nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. So, as I mentioned, I'll quickly introduce the 3Rs Collaborative. And my name is Lauren Young. I'm the program manager from the 3Rs Collaborative, and I'm excited to be hosting this workshop today. So I'll begin with a quick overview of what the 3Rs Collaborative MPS initiative is. And overall, our collaboration across industries makes us a powerful force to forward the 3Rs. And our Microphysiological Systems initiative seeks to increase adoption and regulatory acceptance of MPS in coordination with animal studies. And our initiative includes over 45 member institutions, a total of over 90 individual members. And we have a few major aims. So one, to provide thought leadership and expert consensus from developers combined with other stakeholders on implementation of MPS. To facilitate appropriate discussion, collaboration, and sharing. To create direct engagement between developers and regulatory agencies. To develop external partnerships and collaborations. And to create resources to facilitate increased engagement with commercial MPS developers. And we accomplish this via our three work streams. So our end user interfacing, our regulatory, and then our technology expo, which I'll quickly provide a brief overview of each of these on the next slides. So first, we're engaging consistently with end users. So in 2021, we hosted our first webinar on liver MPS and have hosted 2 webinars and 3 workshops in 2022 and then 4 in 2023. In 2022, we also were at the reception for the MPS World Summit. We handed out flyers and pins for our members. And then in 2023, we actually talked at the MPS World Summit, and then we also at the end joined Altex as a member and had our first Corners publication the beginning of 2024. And you can check out our presentation page here for all of our previous webinars and workshops. And soon this can also be found on the events tab of our website. And in terms of our regulatory group progress, beginning in 2021, we presented a talk at the MPS World Summit and began drafting publications on an overview of regulatory acceptance. We continued this writing in 2022 and then also engaged with FDA, Standards Coordinating Body, NIST, and CAAT, and presented at the 3D Tissue Summit. And then in 2023, we clarified and got consensus for our publication. We were in regular talks with establishing a PPP, as well as on the 3D Tissue Summit on the regulatory panel. And then finally, we're providing education and thought leadership through these initiatives. So in 2021, we presented a poster at the MPS World Summit. We began weekly LinkedIn posts. In 2022, we represented the US on the regional update panel at the MPS World Summit. In 2023, we presented at the 3R Sharing Conference, created a one-pager on the FDA Modernization Act, and presented at the World Congress on Alternatives and ALAS. And we look forward to many more presentations and initiative progress in 2024. And one of our main outputs is our technology hub, which I'll provide the link for later in the chat. But just want to quickly remind everyone that you can submit questions using the Q&A button, and these will be answered time permitting after each presenter or in the roundtable. And you can visit our website to learn more or email me if you are interested in joining our initiative or becoming one of our sponsors.
Hi, everyone. My name is David Kukla. I am part of the IQ MPS affiliate, and I specifically serve on our strategic partnerships and communications team. So I just wanted to give kind of a quick overview of the IQ MPS, but feel free to visit our website that I have listed in the lower left corner, iqmps.org, if you're interested in any more information. Okay. The IQMPS is currently composed of 26 member companies, and we have representatives from drug safety, 3Rs, ADME, and PDPK. Our leadership team is currently made up of Anya Kopek, Rhiannon David, Kim Homan, and Rhiannon Hardwick. And then besides our 2024 leadership team, we also have a steering committee, and this is made up of 2 individuals from each company that's a member of the IQMPS. And then to the right, I just have a little bit more highlights on the IQMPS. Currently, we have over 115 active participants, which meet several times throughout the year. The IQMPS has 3 publications, and it's currently working on several more. And then also we do have a LinkedIn page, so feel free to follow us there to keep up with IQMPS news. So there are 4 main goals of the IQMPS. One is to serve as a thought leader for both MPS developers and stakeholder organizations in the industry, implementation, and qualification of MPS models. Also to provide a venue for appropriate cross-pharma collaboration and data sharing. Additionally, to create focused engagement between both industry and regulatory agencies on the current status and also the evolving field of MPS in an industry setting, as well as develop external partnerships and collaborations to help enhance the inclusion of industry priorities. And to do this, the IQ-MPS is made up of 5 affiliate work streams, and this includes manuscript publications, regulatory outreach, proof of concept studies through pilot project teams, strategic partnerships and communication team, as well as a landscape gap survey team. And here I just have an overview of our external partnerships over the past few years, starting back in 2018. However, besides this, we have several other activities within cross-pharma collaboration and data sharing, regulatory agency engagement, as well as thought leadership. But some of the highlights from our external partnerships team in 2021, as we already mentioned from Lauren, we co-partnered with the 3Rs Collaborative and established an MOU, which allowed us to work together to create workshops or webinars such as this. And in 2022 and 2023, with the 3Rs Collaborative, we had 4 webinars each year on different MPS systems, such as neuro, liver, blood-brain barrier, vascular, as well as GI. And then within 2024, in addition to this webinar, we have 3 additional planned throughout this year on a quarterly basis. And then in addition to that, we recently participated in an IQMPS FDA workshop. And in this workshop, we discussed where and when animal-based MPS may benefit the advancement of the field, as well as establishment of confidence in the predictive nature of these platforms. And we compared the perspectives of the IQMPS and FDA on animal MPS, as well as provided examples where in vitro models are aiding 3R's efforts. But besides this, we're really excited to continue these efforts throughout the rest of this year and upcoming years. So that's kind of all I have right now. But if you have any questions, feel free to reach out or visit the IQMPS website for more information.
All right. So I'm chief scientist and co-founder at Hesperos, along with Mike Schuler, and I'm also a professor at the University of Central Florida. Just give you a quick overview of Hesperos. It's founded by Mike Schuler and I. We've licensed a lot of patents from Cornell and UCF. We won the Lush Prize earlier. We've used a lot of SBIR grants to establish our facility. We now have a 14,000 square foot facility here in Orlando, Florida. We've got about 50 people at the company so far. We're a CRO. We don't offer our services. And we also were the first company to take our efficacy data from one of our models and get a successful IND that authorized a phase 2 clinical trial for a rare disease with Sanofi. Everything that we do is really built around this idea of clinically relevant functional readouts. So, much like you go into a doctor's office, he doesn't immediately stick a needle in your arm to look at biomarkers. He looks, how are you walking? How are you talking? Are you making sense? He checks your reflexes. He might ask about elimination. What he's really looking for is, are your muscles contracting okay? Your electrical activity of neurons and cardiac cells working? Your neuromuscular junctions working fine? Whether or not your barrier integrity is intact. So what this allows us to do is non-invasive look for acute, but more importantly, for chronic monitoring of these human chip systems, but without cell death. We really are trying to do the clinically relevant aspect of it. And allows mechanistic determination as well, certainly for toxicity and target identification. And what I'm going to describe today are systems that have thin layers of silicon where we can put cardiac cells on that, so when they contract, they will deflect laser. We can get force. We can pattern cardiomyocytes over top of electrodes to be able to get conduction velocity and QT interval. We can also look at skeletal muscle. Again, same thing, look at force. We can look at neurons on microelectrodes, either patterned or unpatterned. We can still look at biomarkers. But what we can do, we can combine these together in multi-organ systems where we culture out the cells, okay, on each individual chip. Then we put in their own conditions, then we put them into a platform, and then put down a gasket on top of it where we define a flow path where we can then have the same media flowing over all the different chips, whether it be cardiac, neuronal, skeletal muscle, liver. We can also swap out chips very easy to reconfigure it. Say if we wanted to put pancreas in there, we can. It's all based upon the idea Mike Schuler came up with back in the 80s and 90s of taking physiologically based pharmacokinetic models, modifying them to make multi-organ systems. This is one of our more advanced systems here for looking at opioid overdose and recovery at the same time with off-target toxicity. These are a lot of the papers that fueled this work. And the other major advance that Mike came up with was this idea of a pumpless platform where we get rid of all the tubing, get all the pumps, and it makes it much more easier to operate because we just use gravity to pump everything. More importantly, it also makes it a low volume system. So if we have a competent liver, we can see parent and metabolite in the same system. So we've been able to use that model and to be able to put circulating monocytes in the system. This was detailed in a paper with Roche that was published in Advanced Science in 2022. And what we wanted to do is to show that first, the monocytes weren't activated by simply circulating through the system in the presence of the different cell types, and they weren't. You can see here that the markers for activation are minimal, okay, for a 7-day period. However, we can say hit them with IL-4 and show that they turn into macrophages fairly readily in the system, so we can activate them. So what we wanted to do was to look at whether or not we could create the M1 versus the M2 phenotype. So we selectively damage the cardiac cells, the amiodarone. You see the immune cells then going after the pattern cardiomyocytes, okay? And what we saw here is that the amiodarone damaged the cardiac cells. So you got a reduction in force. But with the immune cells there, it was even worse. Same thing with conduction velocity, was damaged, but actually worse with the presence of the immune cells. Same thing with beat frequency. But skeletal muscle and liver were not affected. So here we have a non-invasive functional readout. So if you want to look what's happening in a multi-organ system in the presence of immune cells with your drug, we can actually give a functional real-time readout, okay, without having to take the system apart. At the same time, we can monitor for biomarkers. In this case, we're looking at IL-6. Now, IL-6 in this particular case was increased and then stayed up, whereas all the other markers for the inflammatory macrophages were not maintained, which suggested to us strongly that we had the M2 or restorative phenotype. Now, we can also take and activate them as well using LPS, a bacterial protein, and so we can get inflammation in the system. So again, we did the same test, same model. You can see with the immune cells, there was no changes, but when we added the immune cells with the bacterial protein and interferon gamma, we saw that the force was decreased tremendously. Now, conduction velocity and beat frequency weren't affected, so the electrical aspects weren't affected. But you can see the force now on the muscle was also affected, and there was also a problem with the liver cells as well. In this particular case, IL-6 went up, but then came down. But then IL-8, IL-10, RANTase, MIP1, and TNF-alpha, and MCP1 were all maintained, showing that we had the inflammatory M1 phenotype. Now, so what we're going to talk about is how we can integrate immune function into our clinically relevant functional readouts. So that's one aspect in a multi-organ system of recirculating flow. We can also do the same thing in our neuromuscular junction model, where what we could do is we can actually take motor neurons and muscle, grow axons through the tunnels to be able to integrate the muscle on the other side. And again, sticking with this idea of clinically relevant functional readouts, what we can do, we talk to somebody who ran clinical trials, say, well, okay, what are some of the tests you have them do? And what they actually have them do a task faster and faster with their hands. What we can actually do is mimic that by stimulating faster and faster and see if it drives into tetanus. And this was a publication came out in Advanced Therapeutics, where we can basically look at the fidelity in the wild type and see that every time we do a stimulation, we get a corresponding contraction. But for SOD1 and two different phenotypes of SOD1 for ALS, we get some sporadic contractions and also partial tetanus. With the other mutation, we don't get tetanus at all. And then with FOS, we get some loss of fidelity and then loss of tetanus as well. We can know that this is actually acting at the neuromuscular junction because we look at the wild type muscle on the other side. We don't see any deficits when we stimulate the muscle directly. And these are statistically significant changes. We can then do the same thing for fatigue index. Again, a equation taken directly out of clinical trials and showing the fatigue index went up for all these different mutations. And what we did is we, working with the Apellis, they wanted to see an immune model of this. So we added microglia onto the motor neuron side and then Schwann cells and either unactivated macrophages or activated macrophages on the other side. You see with the unactivated macrophages in a quick apartheid system, you add in complement serum, you do see a reduction, and you see that their molecule, which is a complement inhibiting drug, that you saw increases as you increase the dose. Now, in the activated macrophages, what we expected was a large decrease in function, but again, you saw some recovery, okay, in NMJ number with their drug. Now, with fatigue index, you do see that with the unactivated macrophages, you don't see much effect, but you do see that in the activated macrophages at the highest concentration, a large reduction, okay, in the fatigue index. And then again, monitoring the biomarker that's been shown to be effective in ALS, which is neurofilament light. So we can still look at biomarkers and correlate the function with the biomarkers in a immune competent functional system. So we can also do the same thing. We have models for long-term potentiation, which is useful for diseases such as Alzheimer's disease, frontotemporal dementia, and others. Again, it's a cartoon I did many years ago where we were trying to look at the hippocampus, imparting information into a clinical layer. So we've actually taken and actually created that system where we and basically reproduce the system for doing dual patch clamp electrophysiology, because we can pattern neurons on top of the electrodes and then hook them up using surface chemistry through their axons, stimulate at these electrodes, and record at these. In fact, the same thing you're doing with patch clamp electrophysiology. Again, we know these are biological signals because we hit them with lidocaine after each experiment, showing that they are abolished, and these are biological signals. This was published in Stem Cell Reports. And then to show definitively we had LTP, we blocked the AMPA receptors, we blocked the ability to do LTP, and the same thing with the NMDA receptors, we blocked the ability to do LTP. And then we actually wanted to then show in the disease models, we took A beta scrambled using patch clamp and showed we could get nice signals from the cells. We shut it off with the A beta oligomers, reversed it with the donepezil, which is an improved A beta drug, and showed direct correlation with the MEA systems. Again, just showing it again without cell death, and then showing that we can actually then, this is just a graph plot of this where we can show we can induce LTP in the presence of donepezil. We did the same thing with tau oligomers, showing with the control that you still get LTP, but it's abolished with the tau oligomers. This is published in Alzheimer's and Dementia. But the key thing is we can use this model to then look at microglia. We put in microglia that is non-activated, and we see as we increase the ratio of microglia, we actually get an enhancement in LTP, which has been postulated that microglia are helping with the information transfer. And again, you can also go too high in microglia. It's a nice sweet spot here. But when we put in the activated microglia, we see that the LTP is compromised as we go to higher and higher ratios. So what I've done is I've shown you 3 different systems where we can integrate functional immune cells into a very clinically relevant readout system to see exactly what the immune cells are doing in a tissue while a drug is being administered to that. And again, here's the contact information for Hesperos. If you'd like to talk to us again.
Thank you, Laura. You're welcome. I will give a brief introduction in the Mimetas immune competent models that we have. The presentation first will start with a short general introduction into Mimetas, and then I will dive into the data in different models that we developed. So Mimetas provides human relevant disease models to study human disease process, and we also aim to accelerate improving the drug discovery process by generating data. And we do that by building our models in different blocks. So we think if you build an in vitro model, you need to consider to implement the epithelial part. You want to have the connective tissue and those supporting cells that are present in this connective tissue. You want to have the vasculature there to have your nutrients at the right place, but also for the trafficking of immune cells. And you want to have your immune cells in there. Next to that, we think it's very important that you also can really scale your platform. So we make use of the organoplates, which has up to 96 chips per platform. And in that way, you can generate a lot of data. Benefit of the platform that it's also automatable and works with different Microsoft setups. So our company is already existing for more than 10 years. We work with a lot of different partners, and the main office is in the Netherlands, but we also have a small lab in the US and office in Japan. Then I would like to dive a little bit more into the models. So we have a lot of different modeling expertise, but today I would want to focus on the gut, kidney, liver, and the vasculature. And I would like to start with the gut. Here you can see a basic immune competent model of the gut, where we use KCO2 cells and HT29 cells to make a tubular structure. You can see it in the center that it's very a good platform for imaging. And next to that, we added immune cells, THP1 cells, and MUD cells into the other lane so that we have an immune competent model. Then we started to induce inflammation by adding inflammatory triggers. And here we used a combination of TNF alpha and IL-1 beta. And we looked at the barrier of the gut HT29 tube over time. And what you can see is that after 72 hours, the barrier is partly disrupted and resulting in a reduction in the tear with inflammatory mediators. If we then add a compound TPCA1, you can see that we can rescue this drop in the barrier. We didn't only use the barrier integrity also readout, but also looked at secretion of cytokines. And you can see that we have an increase in IL-8 release when we add inflammatory mediators, and that we can block this by the addition of the TPCA1. So this is a really nice model, but mainly consists of cell lines, and preferably you want to do a little bit similar, but then with primary cells. And we decided to work with colon organoid cells. And we use this organoids to also generate a tubular structure in our platform, as you can see on the left. And we looked at the barrier integrity of this tube over time, and we could have a reasonable stable barrier for over 10 days of cell culture. If you would want to model an inflammatory disease like IBD, you also would want to include immune cells. So in the second panel, you can see the addition of macrophages in the ECM quite close to the organoid tube. And you can really see they are stained for CD163, so more M2 marker, that they are fully stretched cells. And then we wondered whether we could still push the cells to a more inflammatory state or more alternative M2 state. So we could do that. And on the bottom, you can see a phagocytosis assay where you can see that the M2 macrophages take up much more of the yellow particles than the M1 ones, which are just hardly visible due to that they are more in the phase only visible in the phase contrast imaging and not with the yellow. So you of course can look at the plasticity of your cells, but you can also look at the migration of your immune cells towards the gut. So in the right panel, we have a gut organoid tubular structure in the top, and in the bottom, we have an endothelial vessel, which we perfuse with immune cells that we stained with the fluorescent label. And when we add a trigger, you can see that those monocytes start to extravasate out of the endothelium into the ECM. In the end, not only the monocyte, but also other stromal cells support inflammation in IBD. So for us, the next step is incorporating the stromal compartment. And this is how that looks like for us. On the top, you can see in purple the gut organoid tube, and you see the macrophages are really close by the gut organoid tube, and you have also the fibroblast cells present. And in the end, we now want to look at different inflammatory triggers and how this more complex model responds to this in barrier disruption, but also in cytokine release. If we then look at a more basic model, this I would say definitely far more simple and more easy. It's a vascular model. In this case, we used euphic endothelial cells, did let them form a vascular tube, and then looked at the barrier disruption. We started quite easily by adding inflammatory mediators, and then looked at the barrier real time over time. And what you can see when we add different concentrations of TNF, you see that the barrier is going down. And with the lower concentrations of TNF, it's already rescued after 48 hours. For an interferon gamma, it's a little bit different. If there is a drop, the drop will stay there. In the combination, the drops are more significant. We can then multiplex this with other assays like the expression of adhesion markers like ImmuONE, and we can actually quantify that. And this is still a model looking at vascular inflammation, but without the immune cells. In the end, we also wanted to look at the effect of the immune cells on barrier disruption. So we added T cells in the UVAC tube and then followed the barrier over time. If we just add normal non-stimulated CD3 T cells in the endothelial tube, we don't see any effect on the barrier. On the moment that we added CD3, CD28 stimulated T cells, we see that the barrier is really disrupted in a similar way if we mix the two cytokines TNF and interferon gamma. So this is not telling yet whether we also have migration of the T cells. So we try to stimulate the migration of the T cells out of the endothelial vessel with CXCL12. And as you can see in the middle, with CXCL12, we do get T cells in the middle compartment. So there they migrated into the ECM. And when we quantified this, we saw that both unstimulated and stimulated T cells migrated into the ECM, so that the barrier drop you don't need a barrier drop to get the migration of the T cells out of the endothelium. Now I would want to focus on a kidney migration model. So it's a model that we already have for quite some time, where we have an RP-TEC tube of mimicking the kidney and an endothelial tube of UVACs on the other sides. And in this case, we use this model to add T cells in the UVAC endothelial tube, and then looked at the migration of these cells towards the kidney compartment. At the right, you can see the data of the immune cells directly after addition. And in gray, you can see the number of T cells that start adhering to the endothelial vessel. And you can see that there's an increased amount if you add more T cells, but also if you stimulate your vessel and kidney with inflammatory mediators. If we then look at the culture after 3 days, you see that the attachment is more similar in all conditions, but that only in the conditions where you have stimulation of your model, you also get migration of your T cells into the ECM and into the renal compartment. In this way, you can study renal inflammation and also trying to block it. One of the more drawbacks of our models is that most of our models have bidirectional flow. And what we currently try to apply in more models is a unidirectional flow. Here you can see an example of our vascularized liver, which consists of primary hepatocytes, LDACs, and stellate cells. You can see there's a nice vascular network formed. You have polymerization of your cells, albumin presence. And what we now can do with the unidirectional approach is also perfuse this model with T cells. And you can see here in white the T cells going from one side of the model, perfusing towards the other sides. And for the next step, we want to induce inflammation in the liver and then see how this contributes to different diseases. So in Mimetas, we work together with a lot of companies and academic partners, and in different ways. We do drug development partnerships, we do services for companies. And we also sell our platform and devices that can be used in combination with our platform. Thank you for your attention. If there are any questions, you're free to ask them.
Thanks so much, Lenny. We do have a few minutes for questions. It looks like there is already one in the Q&A. So someone asks, I wonder if you have a validated protocol for co-culturing immune cells, including media conditions and cell source in Mimetas models, and these protocols can be shared with end users, which I think that's a question. Or do you support these experiments primarily as fee-for-service at Mimetas?
So we have a few publications on, or we have two publications on endothelial extravasation of T cells on both UVEX and HMAP1 cells. Those applications we do support. If it are slightly different cells, we cannot fully support it, and we more do fee-for-service work where we do the development.
Great. Another question, how the monodirectional flow is generated and maintained in the plate?
So that's based on the different wells and setups. And so we still use the same rocking platform, so the organoflow. Only by connecting different wells, we can create unidirectional flow through the plate and the vessels.
Great. And a few more questions. Someone says, beautiful data and readout establishments. What were the TEER absolute values in the cell line model? That's one of the questions.
The Keiko HT29, I think it was around the absolute, I don't know. So we correlated to the area, which was around 300 or 400. But for the absolute values, they are much higher. So, sorry, cannot answer that.
No problem. The same person also asked, which colon organoid source do you use?
So for those, we isolate the organoids ourselves. So we did get fresh tissue in and did the isolation in-house.
Great. And I think we have time for one more question. Do you see differences with the type of ECM you use? So collagen, matrigel, mixture of the two.
So for most models, I don't think we use, or neuronal models often use matrigel. Other models use mostly collagens. But definitely we do see improvement for some models if we mix the collagen with, for example, laminin, fibronectin, and other stuff. But awesome. I wouldn't say matrigel collagen.
Okay, so hi everyone, and thank you for the opportunity to present some data here. Today I will talk a bit about our immune competent line on chip and specifically some applications on immunotherapy studies. So at AlveoliX, we are an organs-on-chip company, as some of you might know. Specifically, we provide barrier-on-chip technology. So basically, we've interconnected biology and engineering aspects, and we came up with this platform, which we call barrier-on-chip platform. And this is composed of the actuator and the exchanger, which are two electropneumatic controlling units that are used for controlling the microfluidic channels and the valves. Then there is this docking station, the black one here, which is really a docking station, so playing interface between the controlling units and the consumable, which is here, and it's called AX12, and is where the cell culture takes place. So the AX12 has a 96 well plate format. It has 12 independent units, that's why it's called AX12. And every unit is composed of 3 wells, which are interconnected on the basolateral side via microchannels that are controlled with some valves that are controlled by the exchanger and the actuator. So there are 3 wells, and the inlet and outlet wells are used mainly for medium exchange, for instance, and the central well contains a porous and very thin and flexible membrane where we can culture the cells. This membrane is hanging in the well, so it creates an apical and a basolateral chamber. And since it's very thin, it's 3.5 micrometer, the thickness, you see here representation of a typical co-culture that we do between epithelial and endothelial cells, and you see how close to each other they are. So what we also included in our technology is the mechanical stimulation, and this we do in 3D. So we inspired our mechanism to the breathing concept. So we have a membrane on the bottom of the basal chamber, which can be deflected thanks to the actuator in a cavity that is on the bottom of the basal chamber, or can also be deflected upwards in the basal chamber. And this movement is then directly transmitted on the cell membrane where the cells are cultured. So these cells result in a 3D stretch motion that you see here. For example, for the lung, it would be the breathing motion. However, it's very easy to set different amplitudes and frequencies to then mimic. You can build up basically any protocol that is relevant for any organ of the human body. But today, as I said, we focus on the lung, so we are looking at the breathing motion. So then, in the lung-on-chip models, we also worked quite a lot in the past years with immune cells, so also involving this immune component in our models. And we've worked mainly with PBMC, so peripheral blood mononuclear cells that we isolate and use in-house. And these, as you might know, they are a bit a mixture of immune cells. They include T cells, B cells, NK cells, and also some monocytes and dendritic cells. We also worked with the THP1 cell line, not so extensively like with the PBMCs, and then we differentiated that into alveolar macrophages on the chip. So today I will show you some examples of application with the PBMCs. And another thing that we have to think about when we put the immune cells in our chip is in which compartment we want to put them. So everything is possible. We can put them both on the endothelial side, for example, which is, in the case of aquaculture, it's maybe the most logic or more similar to the human situation because they're on the blood side. However, we can also put them on the epithelium side. When, for example, for your study, the main question would be what happens if the immune cells manage to cross and go in direct contact with the epithelium, what happens there? So kind of mimic the worst case scenario. So this is also possible. So when we first started to have a look into immune cells on the chip, we did some proof of concept studies, and you see here how, for example, with PBMC flowing in the chip, in a chip coated with UVEC cells, they were specifically recruited only in presence of the pro-inflammatory trigger LPS. And we also checked a bit higher magnification to see and to prove that the cells, the PBMCs, are easily going on the endothelial cells, then they can go between the cells and also pass through the pores towards the other side of the membrane. Here it's also one of our first very simple studies that we did on the chip. So we treated with LPS a co-culture alveolar barrier system, so with epithelial and endothelial cells, and we could detect very little or no barrier loss just with LPS. With PBMC alone the same, but when we used the PBMC in the model and then treated with LPS, we managed to have a barrier disruption here. And same for islet secretion, we can see that once the PBMCs are included in the model, the LPS effect is much stronger than the LPS alone, basically. So as I said before, today I will show you some case studies of this immune-competent lung-on-chip, especially focusing on cancer immunotherapy, because this is one of the fields where it's important to have alternative models to animals, because often animal models are not able to give some reliable results because of the differences in the immune system between species. So the first example is IL-2-induced vascular leak syndrome. So for this one, interleukin-2 is actually an approved drug, a cancer immunotherapy, which is approved in high dose for metastatic melanoma and renal carcinoma. However, it is not so widely used also because it's associated with vascular leak syndrome, which in the lungs can lead to pulmonary edema and inflammation. However, this doesn't happen in all the donors that undergo this therapy, but more or less 1/3 of them develop this vascular leak syndrome. So we wanted to try to reconstruct this clinical situation on chip. And for this, we used our patient-derived primary alveolar epithelial cells together with endothelial cells. So it was a co-culture like you see here. And we also had PBMCs as immune component. And when we treated with the combination, so the blue bars, we could see that actually in all the donors that we tested, in this study it was 4 donors, we could see that the PBMC plus IL-2 condition increased the cytokines more or less significantly in all the donors. However, this cytokine storm that we saw led to apical toxicity and also a barrier impairment only in 1 donor out of 4. So we could see, for example, here we called it responder. Just to separate it a little bit, we could see that in this donor, when the treatment with PMC and IL-2 decreased the barrier integrity, while in the other 3 donors, basically nothing happens. And the same is for the apical cytotoxicity. You see here an increasing, well, significant increase in presence of PMCs, and in the other 3 donors, nothing happens. So this data helped us to show that it's possible to reproduce donor-to-donor variability as seen in the clinics, and this is not possible when working with cell lines, for example. So another example case study that I want to show you today is about safety profiling of a T-cell bispecific antibody. So these kind of antibodies are used for cancer immunotherapy, for example, and are antibodies that are engineered for recognizing 2 different antigens on the 2 arms. So in this case, on 1 arm, there was CD3 for recognizing T cells and bringing them close to the target cells, and the target was folate receptor 1. So folate receptor 1 is overexpressed in many types of cancer, for example, ovarian cancer, lung cancer. And the problem of this kind of therapeutics is that this folate receptor 1 is also expressed in the epithelium, for example, in the lung and also in the kidney. So that's why an on-target but off-tumor toxicity is a risk, and during the development, it needs to be tested. So actually, in this study, we used a specific compound that was already used in preclinical studies, and despite the high efficacy in tumor killing in mice models, when then the molecule went to monkey studies, it was stopped. Actually, the clinical development was stopped because of respiratory inflammation detected in the lungs, especially of the monkey, and also electrocyte infiltration. So we tried to reproduce this same toxicity on the chip, on human cells, to try to see if we are able to detect with our model in vitro the same toxicity. So we used again the patient-derived primary epithelial cells, this time in monoculture, and the immune cells were again the PMCs. So we had this compound that I explained you before, folate receptor 1 TCB, and the negative control was a non-functional variant of the same format of TCB. We could see here that when we introduced the TCB, always with PMCs on the chip, the highest dose led to a loss of barrier function, and also to a significant increase in pro-inflammatory cytokines release. We then checked for cytotoxicity with by live imaging of Caspase-3-7, and we could see, when we quantified one of the time points, we could see also here a dose-dependent increase in apoptosis, and as well, when we checked for cell death, so at the H release, we also saw significant increase given by the high dose. We also had a closer look to the immune cells on this system. So we first, they were tracked. So we were able to also follow them live on the chips. And what we did was at the end of the experiment, count how many of the immune cells were still attached to the epithelium. And we could see that specifically, they were specifically recruited to the epithelium in presence of the TCD. We then harvested the immune cells from the chips and did some flow cytometry analysis on these cells. And also here we could detect some CD4 and CD8 T cells that were activated by the high dose significantly, and also some of the CD8 were degranulating. So actually, with this data, we showed that this model can predict the toxicity similar to what was found in monkeys studies, and therefore this tool is very useful for helping in decision making. In preclinical decision making, and also just to give you an idea of the impact that this data can have, these data were already used for 5 IND submissions. So just to conclude, I showed you here some data with the AlveoliX lung chip with immune cells, which brings certain advantages, such as, for example, the synergistic effect. So I mean that in order to see certain effects, you need the immune component. Also, they allow an accurate inflammatory response recapitulation, and also immune-specific readouts, like the checking for differentiation or activation of the immune cells. And also an advantage is for sure that it's human-specific, so you don't have the problem of the different immune system between different species. And some of the applications, like I showed you today, is the cancer immunotherapy is one of the main fields. But of course, they can be used for inflammatory diseases in general and for safety assessment and toxicity to check for new variants of molecules. So thank you all for your attention. I'm happy to get any question now or later, whenever you want. Thanks
so much, Julia. We do have a minute for a quick question. Are PBMC and epithelial cells donor matched? Is it necessary to take this into account? That's a good question. So it depends on the application we do. But for example, in the data I showed you, they were not matched. But in some other data, we already did some studies where we really tried to match the PBMC and the epithelial donor HLA typing, not exactly coming from the same donor, but from at least the same HLA typing. And it's also something that we think about to try to get the same, from the same donor, the two cell sources. Yes.
Hi, so my name is Lewis. I am the commercial leader at ImmuONE at the moment. And this talk is about our models, our immune competent lung models, and how we've used them to profile alveolar macrophage responses to inhaled compounds. So a little bit of background to why alveolar macrophage responses matter. Ultimately, the alveolar macrophages are the first responders. They're the first cell that's going to come into contact with any inhaled particle. They're going to have key roles in particle clearance, homeostasis, resolution and initiation of inflammation. And it just makes sense to have them in a model of the lung to assess lung safety, as there are really limited endpoints you can assess with just using 2D epithelial cell culture. You could just have tear or like IL-8 production, but that's not really going to give you much indication on the identifying the harm that could be caused to the lungs, even if there's not obvious toxicity there. So yeah, a little bit about macrophages there, and why we've kind of included them into our assays. And what we do here at ImmuONE is we do three things. Let's say we have three arms. We have kind of our models that we produce that are commercially available. So they are immune-focused cell culture models that center around the alveolar macrophage. We have two that are commercially available at the moment, which are ImmuPhage and our ImmuLung, and I'll talk about them in a minute. We also do a lot of like academic assessment strategies. So as a contract research organization, we work with a lot of clients that have all sorts of weird and wonderful problems, all the way from the nose to the alveolus and sometimes even beyond that space, where we provide academic proposals in order to meet the variety of problems they may have. A lot of that time it's correlating to in vivo data. And third of all, we have novel assessment approaches that center around multi-endpoint analysis and high content image analysis. And it is that which I will predominantly talk about for the latter half of this talk. So in terms of our models, we have started off having kind of a layer by layer development. So assembling platforms of models with increasing complexity so that we can answer urgent questions. And again, you can make a model as complicated as you'd like, but sometimes that makes it more costly. It can make it more difficult for the end user, and it can sometimes not be really what you need to answer that problem. So what we have with our models, we have kind of our the base level, which is the ImmuPhage, so our alveolar macrophages and monoculture. We then build it up to have an ImmuLung, which is our type 1 or type 2 alveolar epithelial cells and co-culture with macrophages. We're then in our R&D stage at the moment, and we are hoping to get out within the next couple of years our kind of ImmuLung FT, which is with the fibroblasts as well, in a kind of triculture setup, and then eventually working towards a fibrosis model of the lung. So all of that to come, excitingly. So just for today, I'll give you a bit of background about the two models, the ImmuPhage and the ImmuLung. So the ImmuPhage is an alveolar macrophage cell line that we have developed in-house. And retain some of the features of a alveolar macrophage cell. So for example, it is non-proliferative, it is terminally differentiated, as can be seen on this blue line here, where we have viable cells on the y-axis against the time in days on the x-axis, and we can see from undifferentiated monocytes or existing protocols, the number of viable cells increases over time, but actually we with ImmuPhage, that is terminally differentiated and will not proliferate. In terms of phenotype, we've done extensive phenotyping of this. I've just got a few markers on here, so CD14, CD11C, and CD11B low, both plus and minus LPS, just to show some of the phenotypic characteristics of the CD markers expressed. But again, there's more CD marker data if anyone's interested. And again, phagocytic function of the ImmuPhage compared to kind of existing protocols or undifferentiated monocytes, they have ImmuPhage has a much higher phagocytic capability or capacity, with around 80% of cells able to undergo phagocytosis, and a standardized response to challenge. So we've chucked all sorts of nasty stuff on these cells. And in terms of amiodarone, so a known phospholipidosis inducer, you will get increases in intracellular phospholipid content at both day 0 all the way up to your 5 weeks in culture and beyond. And the same with staurosporine, which is fairly nasty. If you treat with staurosporine, you will get cell death at day 0 and all the way up to day 35. And so those are just our alveolar macrophages. For the co-culture model, this has the alveolar epithelial barrier and immune competence. So it will have a fairly decent tier value that is maintained from about 10 days in culture onwards, both at the liquid-liquid interface and air-liquid interface. It has functional tight junctions as well, so there's some kind of low apparent permeability down the bottom, yet whereas you treat with something like trypsin EDTA, you're going to increase that permeability across and destroy those tight junctions. It is immune competent, so if you treat with LPS, surprise, surprise, you'll get changes in cytokine presence, so increases in tumor necrosis factor alpha, IL-8, IL-1 beta, IL-6 as well. whereas you're getting much lower levels in just your epithelial only model. And finally, we have, like I mentioned with the ImmuONE, we've treated it with all sorts of stuff over the years, so various stimuli, so that we have a database of kind of known compounds and how we expect our models to respond. Everything from amiodarone, saurosporine, to things like cigarette smoke, LPS, and a range of kind of small molecule modalities, and kind of much larger biologics as well. And we've got a little bit of that data on here, but if you'd like to find out more about it, I've got the some QR codes on here, and I can send some of this over your way at the end if you're interested in learning more about the models. So I guess in terms of the application of these models, when we first started ImmuONE almost 5 years ago now, the company was spun out over a problem that was arising, and that was the concern of immune responses in preclinical studies, and was ultimately one of the reasons why many inhaled compounds were due to fail. And that was because there were these highly vacuolated foamy alveolar macrophages observed in the preclinical studies, and it posed safety concerns, despite companies not knowing the correlation to human, or to actually observe, would this be something that would be found in human cells, and what was the relevance for humans. So just some of the in vivo histopathology findings here, so macrophage accumulation, and these hypertrophic vacuolated macrophages, and then some more of some in vitro images here. You can see it really nicely in this figure B here, where you have your alveolar macrophages, and you can see these really these areas of high vacuolation. So that was being found. So there was all these compounds being withdrawn. So the idea was to fix the problem, do something about it, provide a tool that could allow the identification. So we designed a high content image analysis tool to look at morphology and lipid endpoints for inhaled safety assessment, with endpoints that would correlate to in vivo observation. So simply cells, fluorescent probes, imaging, and image analysis, and what you can see is changes in the morphology and lipid parameters. Now, there is a really interesting paper that was published from our group back a few years ago, but actually when you're looking at macrophage responses to, let's say, amiodarone, that phospholipid inducer, if you look at median values on their own, you're not going to see any significant changes. It is really important to look at the elevated characteristics, the elevated cell characteristics, and look at that population data on a single cell level. And as you start to look at the elevated parameters, you're going to see changes that would be noticed with many of these drugs. Because we're looking at we're looking at changes in the percentage of the population, and actually you're going to miss this if you're just going to be looking at the average data here. So when you start to look at your average data, you have your multi-parameter profiles, we can see that there are changes in phospholipid content and cell death markers. So I'll talk you through some of these graphs here. So this is the percentage of the cell population here on the y-axis, and these are the different parameters here. So for example, parameters 1 and 2, these are the healthy normal parameters, so healthy population, normal nuclear area, and then you start to see elevated parameters here, so elevated nuclear cell area, activity, mitochondrial activity, membrane permeability, all the way to your vacuolation and phospholipidosis parameters. And with your untreated control, the black line here, most of the cells fit into the first two categories, with very few of the cell population, a small percentage of the cell population actually having elevated parameters. Yet when you treat with amiodarone, you can really visibly see that actually there's increases in the cell population that have, let's say, elevated mitochondrial activity, and even this area here, elevated phospholipid content, with this being much more pronounced at the 48-hour time period. So multi-parameter analysis of all of these characteristics means you don't just have to look at one endpoint as well. You can look at many different endpoints at the same time, and you can identify the macrophage responses to many known phospholipid fibrosis inducers. And so I guess in the next part of this study, I'm going to talk you through some data that was undertaken in our labs where we had a variety of inhaled modalities, some of which were known foamy macrophage inducers and others were safe, the safeties, as I call them. We had rat and human alveolar macrophage models of our immuphage, and then we would treat with our compound, stain with our dye cocktail. That's usually mitochondrial activity, nuclear stain, permeability, neutral lipids, phospholipidosis, undertake high content image analysis, and then look at the morphometric and cell health parameters. There's a little bit of a background about some of the safe and non-safe. So the safe, mainly beta-2 agonists or steroidal anti-inflammatory drugs, and then some of the foamy macrophage inducers were known cationic amphiphilic drugs, or maybe even in some cases pro-apoptotic compounds. And just so you can see some lovely images of it, because I actually find the images quite lovely to look at, some control macrophages, and then areas with highly vacuolated macrophages and fairly swollen, just pretty unhealthy macrophages here in panel C. So let's look at some of these graphs again. So our non-foamy macrophage inducers, most of the cells, if they are untreated, they have these normal cell counts, normal cell health, and very few changes to elevated parameters. And that was kind of what was observed for the non-foamy macrophage inducers. Again, maybe the odd kind of change with some of them in phospholipidosis, but in general, not many changes to the elevator parameters, yet even visibly you can see here with the foamy macrophage inducers, we have many changes to permeability, to the phospholipid content, to the area, the vacuolation profile with these unsafe compounds, and that's even more pronounced at the 48-hour time point here. So I guess the take home from this is you can use this analysis and it can identify that the rat alveolar macrophages are changing in both their phospholipid profiles and their morphology. Again, some human alveolar macrophages, so some fairly normal ones here, and then even this image here, you can just see how highly vacuolated there are. Some really beautiful images there. So yeah, when we look at human alveolar macrophages, our non-foamy macrophage inducers, there's very little changes to elevated parameters. Yet when we look at the foamy macrophage inducers, some of them are affecting the cell health, cell count, and elicting changes to, let's say, permeability and vacuolation, and changes to the nuclear to cell area. And again, 48 hours as well. But for those of you who are fairly eagle-eyed, maybe have noticed that there were the differences between the human and the rats there. The rats actually are that much more sensitive, and probably over 10 to 100-fold more sensitive than the human alveolar macrophages. So I guess just to summarize this, with 18 seconds to go, we've got our two models, the ImmuPhage and ImmuLung. They're immune competent animal-free tools, and can be used for assessing inhalation safety. We use high content image analysis to differentiate between safe and unsafe responses in macrophages, and we can use multi-parameter tools to predict the safety of inhaled medicines and correlate that to in vivo findings of the pathology. And we can even work with rat ex vivo, but yeah, ultimately not wanting to, we want to move away from that and try to work with maybe rat in vitro and the human cells in order to make that transition towards the animal-free model. So again, thanks so much for listening to my talk, and yeah, happy to take any questions.
Okay, thank you so much. Hello everyone, my name is Gautam Mahajan. I lead the field science team at ImmuONE. Today we are going to look at lymphoid follicle chip, or the lymph node on chip. So just to give a brief introduction of ImmuONE's product offerings, so there is an instrumentation module, which is the cell culture module called the SOE, and there's an orb which helps support that. And then there are biokits, which comes with the chips, the pods. Pods are the reservoir for the media and with the cells as well. And the third offering is the basic research kits, which just comes with the chips and pods and enables users to use their own cells. So using those instrumentations and chip and pods, there are different ways to build immunocompetent model. One of them is to have resident immune cells in the chip, such as our quad culture liver chip, which has spoofer cells to model immune-mediated toxicity. We also have an application for adding circulating immune cells. We use that approach, adding PBMCs to model the IBD application on the chip. Both of these have been presented in various formats here. So if you have any questions on them, please feel free to reach out, and we can happy to have a discussion on that. So the other model is the immune cell model on the chip, which is called the lymphoid follicle chip, which is used to evaluate vaccine efficacy. And that is what we are going to focus on today. So first, I would like to walk you through an example of how this chip and the module was used to create lymphoid follicle chip, which aims to improve the vaccine development. As we all know, that vaccine development is a complex process. From discovery to license, it takes billions of dollars with an average 94% chance of failure. Organ-on-chip, they offer a unique way to improve this process by including human relevant cells to avoid this species translation issue. Also, additionally, these organ-on-chips also help capture donor variability in antibody responses to vaccination, which is more suitable to predict these vaccine responses. So with these models, the researchers were then able to test lymphoid follicle chip and see how it responded to antigens. They found that those structures were made under flow and shows the characteristics towards functional germinal centers, closely replicating the in vivo-like functionality. They were also able to recapitulate the human vaccine and adjuvant responses and captures the donor variability in antibody responses to this vaccination. On the right-hand side of the screen, you are seeing one of the publications which were published using this platform and which developed the lymphoid follicle chip. This was like part of my postdoc work as well. So on this screen on the left-hand side, you are seeing a cross-section view of the model with the bottom channel containing B cell, T cells, and monocyte-derived dendritic cells in a hydrogel to form a lymphoid follicle chip. And on the right-hand side of the screen, we are seeing a typical chip after a few days in culture under static versus flow condition. And we can see like how the matrix is reorganization in presence of the follicles and how these follicles are actually generating in the presence of flow. So in the previous slide, we saw a qualitative analysis of these follicles formation between flow versus static condition. We also observed statistically significant differences between the follicle number and the volume between the flow versus the static condition. We also detected AID expression in the flow exposed chips and a correlation between the follicles and AID expression. Just taking a step back, AID expression is important because it mediates the critical antibody class switching, which happens in germinal centers in lymph node in vivo. We also investigated class switching and plasma cell differentiation by using SAT antigen to mimic the presence of dead bacteria on adapted immune responses. So we observed statistically significant expression of CD138 between the unstimulated versus the SAT stimulated condition. CD138 is depicting the plasma cell differentiation here. And we also saw that the formation of CD138 plasma cells are more focused in the large clusters in the follicle areas versus the single cells which were present on the chip. On the right-hand side of the slide, you are also seeing CXCL13 produced by this chip after 8 days in culture and after exposing to SAT. And CXCL13 is a key requirement for lymphoid follicle assembly, and it's often used as a biomarker to predict the lymphoid follicle formation. So we have seen until now how the lymphoid follicle chip is being formed, lymphoid follicles are being developed, class switching, plasma cell differentiation, and now we are looking at how to use this chip to test antibody responses to evaluate vaccines. So we tested H5N1, which is a research-grade antigen, and an adjuvant called squalene in water emulsion, and we observed statistically significant differences between the follicle number between the H5N1 in presence and absence of this SWE. This whole work was just 4 donors of different B cell, T cells, and dendritic cells were tested in this work. So the next step was to look at the antibody production. So we also looked at in presence of this adjuvant, this squalene water emulsion, it leads to increased levels of antibodies against this flu antigen or H5N1 antigen. And the number of replicates between 2Ds and chips, there was statistically significant increase between the levels of secreted antibodies and also the number of chips responding towards this antigen. We also showed a significant enhancement in the number of CD138 plasma cells, which you are seeing on the right-hand side of the slide. So in addition to testing the research-grade material, we also evaluated responses of commercially available vaccines. So we tested a commercially available vaccine called Fluzone, and we tested 8 different donors against that. So at day 9, after exposure of these vaccination, we saw all 8 donors responding to this vaccine, but we also were able to see 2 patient subsets, a high responder group and a low responder group. And this was done in relative to the taunts and controls, which we were able to obtain from different patients. In the middle of the slide, you are looking at the impact of dendritic cells. So we also wanted to see, like, in addition to B cell, T cells, if we add dendritic cells or if we don't add dendritic cells, what does the responses looks like? So we can see here that the amount of antigen antibody produced against this commercially available vaccine, in presence of dendritic cells, there were higher percentage of that antibody produced. And same pattern was shown for CXCL13 as well. In addition, we also saw a strong correlation between CXCL13 responses and the antibodies which were produced by this chip. So the next step for us was to look at the levels of different cytokines which are important for T cell expansion, survival, and helper function. So we looked at interferon gamma, IL-10, IL-2, and GM-CSF, and we compared it against the serum which we obtained from a human volunteer vaccinated with influenza vaccine. So we are seeing here that on-chip interferon gamma and IL-2 showed 8-fold increase upon vaccine exposure, and IL-2 showed 4-fold increase. From serum levels of human volunteers, we showed 4-fold increase for interferon gamma, IL-2, and IL-10. In full transparency, we saw some increase in GM-CSF on the lymphoid follicle chip, but there was no change in serum sample. It might be some background noise coming from GM-CSF being present in the media for monocyte to dendritic cells differentiation. Just on a higher level, some summary here. This chip is based on the use of peripheral immune cells, B cell, T cells, and monocyte-derived dendritic cells, which helps avoid species translation issue. It also showed functional germinal centers similar to those found in secondary and tertiary lymphoid organs in vivo, showing more lymph node-like functionality. And it also were able to recapitulate human vaccine and adjuvant responses, such as 3D follicle formation, expression of AID and CXCL13, plasma cell formation, antigen-specific antibody generation in response to multiple antigens. And it also showed production of clinically relevant cytokines. We were also able to capture donor variability in antibody responses to vaccination. Thank you so much for your attention, and I'm happy to take any question. I'm hoping I left plenty of time for that.
Great. Thank you so much, Gautam. And we do have time for questions, which is great. So the first one, did you test an ECM solely based on collagen and or Matrigel? Why did you choose a mixture of the two in the final model?
Yeah, so the iterations were developed for various ECMs were tested, and we ended up going with this particular mixture of Matrigel and collagen. Because at the end, the mechanical properties coming from this matrix is very similar to the mechanical or the physical properties of lymph node as well. That's why we ended up going with this particular ratio of collagen and Matrigel and media.
Great. And have you tested the affinity of these antibodies detected? Are they as high as animal-produced antibodies?
We did not compare it to the levels of antibodies produced by the animals. No, we did not. Our goal is to move towards more human relevant approach. As you saw, there is 94% chances of vaccine development failure. There is no point of going back and look at what animal models were doing at that point.
Great. And you might have touched on this a bit already, but is it the alignment of ECM in this case that favors the clustering, or is it the flow? What I understand here is that ECM alignment is flow mediated, so it's hard to decipher the most critical element for clustering.
Yeah, so cluster formation, as you saw between the static versus flow condition, you are able to see some clustering or follicle formation in static condition as well. But flow triggers that in a way that there are better encapsulation of those follicles. So flow does play a big role there, but also we need to keep in mind that this is the media is only flowing through the top channel, and the lymphoid follicle chip is the matrix is in the bottom channel. So this is more of a suprafusion approach. So there is a gradient of nutrients travel from top channel to bottom channel as well, which is developmentally very relevant. And that might be the reason we are seeing some of the follicle formation in the flow conditions, but it does need more testing on that.
Great. Do you think there's a golden number of DCs to be added, a minimum below no increase, and a saturation number in terms of B cell percentage, for example?
There has been a few iterations of how much dendritic cells being added to the chip. Personally, I have not seen a case study which customers might be doing it. I have not seen a case study where someone has directly looking at the ratio between B cells and dendritic cells. In this work, which I showed today, there were 2% dendritic cells added to the chip. There has been few iterations which might be in public format. I'm not sure, but I have seen people increasing that number to 10% as well. That's the maximum which I have seen, but I'm not sure if you do increase it any further, does it make it better or worse? It does need some more optimization there.
Awesome. And we have time for one more question. Do you see a correlation between IgG generation and cytokine production?
I can't say if there is a direct correlation there. Cytokine responses in various case studies, not just with the one which I presented today, but which might not be in public format yet. We have constantly seeing cytokine upregulation or changes, exposure to different vaccination for all the donors, but not all the donors are responding to show the antibody production as what you at the same levels as what you saw today in the slides. So it's hard to say if there is a major correlation. But we did see correlation between antibody produced and the follicle formation, and then the CXCL13 expression as well.
Super. We are an organ-on-chip company placed in Italy, in Genoa. And basically, one of the most important application and validation we performed regarding our organ-on-chip is on immuno-oncology, since there are still many limitations behind the development of the immunomodulating agents, and there is also a challenge regarding the fact that immunomodulating agents do not work in all patients. So we have developed an organ-on-chip platform which enables to simultaneously culture three-dimensional tumor models and eventually patient-derived biopsies with immunocells in circulation. So one of the most important benefits of this approach is that the immunocells of the patients are really placed in a physiological flow condition like in the patient. Moreover, we can properly control the fluid flow using an external pumping system and investigate the crosstalk between immunocells and cancer cells and testing the efficacy of novel drugs. So basically, the ImmuONE chip we have developed has been tested using different types of tumor models, thanks to the flexibility of the platform. So the chamber is compatible with the commercially available inserts. Therefore, scientists can culture tumor cells in a monolayer, or eventually work with spheroids, organoids, or matrix-based three-dimensional tumor models, or patient-derived biopsies. And simultaneously, the PBMC coming from the patient or commercially available cells can be placed in circulation. Regarding the tumor model, we have optimized in the last years three-dimensional matrix-based models, because we do believe it's really important, especially for cancer disease modeling, to keep the real heterogeneity of the tumor matrix and tumor microenvironment itself. Therefore, the over-miniaturization might generate unpredictable results. And therefore, we have patented also an alginate-based matrix where cells, tumor cells, display proper cytoskeleton organization, which is also a mirroring of their aggressiveness. We have worked with different types of tumor models, meaning breast cancer, ovarian cancer, pancreatic cancer, and neuroblastoma pediatric cancer. This matrix is optically transparent. Most importantly, it's also stable over time. Therefore, you can also control and carry out long-term experiments for not only short-term drug efficacy assays, but also long-term assays. The stiffness is tunable, and therefore you can set up the condition for the proper migration of cells. And most importantly, it is animal-free, and you can collect the cells at the end of the experiments for further investigation. When you use the neuroblastoma tumor cells, the cells display the proper viability, proliferation, cluster reorganization in the matrix. Most importantly, display the proper reduction of the viability after the drug treatment in a dose-dependent way. And therefore, we went on along the characterization of the tumor, and we double-checked the proper up and down expression of the receptors which are the target of the immunotherapies. For both neuroblastoma cell lines, we nicely observe the downregulation of the PVR and also the upregulation of B7H3, PD-L1, and PD-L2 that are the most important and studied receptors also among many pharma companies. And interestingly, this upregulation was in some cases never observed before in 2D, confirming as a take-home message the key importance of the three-dimensional culture, especially for cancer disease modeling. Similar effect and similar predictive effects were observed also changing the type of the tumor, where, for instance, we co-culture tumor cells with a natural killer, and nicely we observed the increasing of the tumor cell apoptosis. But we decided to increase the complexity of the experimental setup in order to have a setup more similar to the in vivo condition. Therefore, we culture the tumor models into the organ on chip, while immunocells were placed in circulation, and we set up a velocity of the flow like the capillary blood velocity by using the pumping system you see in the picture. Similar experiments were carried out even in advance, also culturing circulating tumor cells, and we successfully demonstrated that CTCs are able to decode the shear stress induced by a physiological fluid flow, changing their viability rate and also their cluster behavior, which is cross-correlated with their aggressiveness. In this case, we used the feature of the organ on chip regarding the possibility to sampling the media in the both tumor compartment and in the bloodstream compartment, since these two compartments are physically separated by a membrane, which can be also eventually functionalized and cellularized with an endothelial layer mimicking the vascular barrier. Therefore, when we sampling the media in the tumor compartment, we quantified the amount of natural killer placed in circulation, and we found a percentage of natural killer extravasated, which is in line with the clinical data. In particular, about 2% of immunocells placed in circulation were able to win the gravity, leave the flow, and going up in the tumor compartment. We had two important internal control. That was the negative control, meaning the absence of the tumor. And interestingly, in this condition, no significant amount of immunocells were observed extravasated. The positive control was the same polymeric matrix without tumor cells and with an increasing of the serum, which is an important chemoattractant. Well, also in this case, the amount of natural killer extravasated was significantly lower than the condition with the tumor. So the take-home message is that this organ on chip was able to successfully recapitulate the tumor-specific immunocells extravasation. Moreover, since the organ on chip is not sealed and therefore the end user can open the lid and collect the tumor sample, it was possible to analyze the tumor matrix and the tumor tissue itself. And with the specific markers, we found immunocells even infiltrated the tumor matrix. So immunocells were able not only to leave the flow and going up through an extravasation, but they were also able to infiltrate the matrix, migrate, reaching the tumor cells. Then they induced a significantly higher apoptosis of the subpopulation of the tumor cells. So this is the confirmation of what I said before. So this matrix enabled to collect the cells, meaning to decross-link the matrix, collect the cells, sort the cells for the CD45 negative population, meaning the tumor cells, and quantify the amount of dead cells that was significantly higher as effect of the activity of the immunocells. Vice versa, when we sorted for the CD45 positive cells, meaning the immunocell subpopulation, we were able to investigate the expression of the CD16 marker, which is a marker of the aggressiveness of immunocells, of natural killer cells. Unluckily for the patients, but luckily for the science, because this data is in line with the clinical data, we found that the subpopulation of the immunocells able to infiltrate the tumor was less CD16 positive than the overall population. And this is the reason of why, for this type of tumor, the immunocells are not so effective. But for sure, these results open the path towards a better and deepened investigation of the interplay between immunocells and tumor cells, and also for testing the efficacy of new immunomodulating agents. And in particular, you can also place in circulation together with immunocells drugs, as we did in this paper, where we placed in circulation an anti-chemotherapeutic drug, and we observed the capacity of this drug to diffuse properly the three-dimensional tumor matrix and induce a tumor cell death. In this slide, you can see that the untreated tumor matrix displayed a nice distribution of proliferating cells positive to Ki67. After dynamic application of the drug, very few proliferating cells were observed, and the drug was able to induce the proper death of cells in a homogeneous way within the three-dimensional matrix. Vice versa, when the same three-dimensional tumor model was cultured in steady condition, still many proliferating cells were observed, and the dead cells were found only in the external part of the matrix. So our hypothesis was that the issue of this unpredictable static result was related to the unpredictable drug diffusion study condition, and we also used the computational fluid dynamic simulation to confirm this hypothesis. Indeed, you can see in the blue line that especially when you use the three-dimensional tumor models, it is mandatory to have a dynamic fluid flow mimicking the vascularization of our tissues for properly diffusing molecules, in this case drugs, and having results in line with the clinical data. In the graph, you can see in the black bars the regression of the tumor in mice model, and in the green the regression of the tumor in our organ on chip. While in gray you can see the same results in terms of tumor regression in steady condition. In this case, you can see unpredictable results, while using the organ on chip, we obtain the same regression as in the animal models, but with, of course, a cruelty-free approach, and also with a faster reduction of timing. The organ on chip we have developed and industrialized enable also to fluidically connect multiple organs. This is especially important in cancer because you can finally recapitulate the overall journey of tumor cells that might leave the primary tumor mass, enter in circulation, survive under physiological flow condition, and eventually metastasize in another tissue. This is what we have done, for instance, by co-culturing breast to bone tissue, and it has been used by many scientists for testing a new there are also different approaches used in our R&D team, but also by other end users and customers. That is the use of patient-derived tissue explant in combination with patient PBMC, and opening the possibility to test different therapies and selecting the optimal one for the specific patient and tumor. What I've decided to show today is what we have done in terms of validation regarding immuno-oncology application, but immunocells are extremely important in many other diseases. And therefore, last take-home message is that we have also in our company other type of chip platforms, like the gut-on-chip platform, where we co-culture the different type of intestinal models with immunocells and eventually also with the bacteria compartment for studying inflammatory diseases like IBD word family, and also for studying different crosstalk between different organs. This is what we have done internally, but as I mentioned, we already have many other applications of this approach by different stakeholders. In some cases, these results have been carried out in their laboratory. In some cases, we work together, and in some other cases, our R&D team carried out this test for them. Thanks for the attention.
Okay, so I'll start with one question that looks like it's for everyone. So the question is, how do you emulate the true blood flow and lymphatic flow that are essential for biomimetic design? So maybe I'll start with James. We'll just go in order in terms of presenters.
Right. Can you hear me okay?
Yep.
Am I crackling again, or is that going away?
You're still a little bit crackly. I'm not sure maybe where your mic's located on your computer, but if you don't have any other options, it's okay.
All right. Sorry, everybody. Teams is always an interesting adventure. But so since we actually do multi-organ flow systems, we can actually use computational fluid dynamics to actually control our flow, control our shear stresses within the systems. We also do pharmacokinetic, pharmacodynamic modeling of all of our systems, and have published the ability to take the modeling, combine it with our on-chip data to be able to do in vivo predictions. So we can do a combination of in silico, in vitro, to be able to answer those questions.
Great. And then Lenny, I'll go to you next.
Yes, so we use a rocker platform, so we cannot reach very high shear. So things like in an artery, we won't reach up to, that's around 15. We can do lower till around 10. And then we do a calculation which degree of rocking we need to do for how many minutes. And that, yeah, we calculate before applying, and if we test 2 or 3 combinations.
Great. Julia.
Yes, so in our case, right now we don't have a perfusion system yet in our system. We are working on that. It's in our pipeline. For now, we really focus more on what happens when there is the direct contact of the immune cells with the epithelial cells or with the endothelial cells. So for now, we've been focusing more on that. But yeah, I agree that it's one of the factors that is relevant for sure. And yeah, that's why also we are working on that.
Great. Lewis?
Yeah, so my answer is probably going to be quite similar to Julia's. Yeah, absolutely. There is a lot of merit in that. But yeah, the perfusion and yeah, incorporating flow into our models is something that's probably a few years beyond. And really at the moment, our focus is developing the high content image analysis stuff, but we are working with partners to kind of get something up together. But yeah, I wouldn't like to comment too much.
Great, thanks. Gautam, I'll go to you next.
Yeah, so MLET system uses a module called Zoe here, which is a pressure-driven system. It's a unidirectional flow with the flow being tunable. So you can go from 0 microliter per hour to 1,000 microliter per hour, depending on what model you are using, what application you are using. And based on the application, something like immune cell recruitment, we run that at higher flow rate to increase the shear forces as well.
Awesome. And then Sylvia.
Yeah, we have a peristaltic pump which generates the velocity and the flow rate, which is cross-correlated to the setup of the pump and the dimension of the tube. So therefore, we can really use a wide range of flow rate and velocity. And we have indeed already studied the behavior of cells in circulation, either tumor cells or immunocells, double checking also that immunocells are not pre-activated in advance, do not adhere to the walls of the tubes, and eventually we can introduce an endothelial layer of endothelial cells generating the barrier which separates the bloodstream with the tissue compartment.
Awesome, thank you. So we do have a few questions remaining for Gautam from Emulate. So I'll start there, and then if the other participants can put any Q&A questions in the box, and please specify if they're for someone specific or for the entire group. So Gautam, there's a question here. Do you know if the donors that were selected for your study have been previously vaccinated with Fluzone or any other flu vaccine?
Yeah, so my best guess is that it's a flu vaccine study. So I'm pretty sure that either these donors were exposed to flu or to the flu vaccine. So what we are seeing is most likely a memory responses. But not in this publication, but if you go on our website, you will see presentation from other users where you can see the responses on naive vaccines as well, something like KLH, which is very basic, and also towards rabies vaccine as well, which is more naive responses. That presentation was done by GSK. It's available on the website, but for that particular questions, most likely these are exposed to flu or to flu vaccination.
Great. I'm just scrolling back through, and there was a question from Lani actually that I didn't get to before. So Lani, this question says, what motivates you to implement unidirection flow? Did you see positive or negative effects on bidirectional flow, and can you give examples?
So I think for simple co-culture models or simple models, the difference are quite minor. If you're going to look at the vasculature where we do co-culture with parasites or smooth muscle cells with endothelial cells, we see that the culture is more relaxed and more stable over time if we have unidirectional flow compared to bidirectional flow. So I do think it's really dependent on the model and the process that you want to mimic, whether it's important or not. But in some cases, definitely yes.
Great. Gautam, there's another question for you here. Any concern in normalizing flu response against tonsil samples, which is usually pediatric and can have less developed immune memory?
So I can't comment on where the tonsils were sourced, but in terms of why we used it is to find the difference in the responder groups. And for that purpose, the samples we chose serves the purpose for that. For that particular analysis, I'm not concerned of the tonsil samples which we isolated or we obtained, but it should be, the normalization should be based on the application you are running. So if you do think your application needs a particular subset of donors, I think we should be aiming for that.
Great. And one final question for you, Gautam. What's the maximum number of cells your chip can support? For how long can you culture the cells in your lymphoid follicle chip?
So two-part question. I'm assuming the first part is also on lymphoid follicle chip. So there is this iteration which I presented today. This lymphoid follicle chip is a high density cell culture module where there are close to, I think, 1 million cells in the chip. After cell seeding, obviously the seeding density are a lot higher than that. But it can, the second part of the question, the longevity, this analysis in this paper has been done for 14 days in culture. But again, there are some work done by other users where they have gone up to 3 to 4 weeks in culture. The other part is the coming back to the cell density part. There is other iteration of this model as well, published. I think it's in a preprint from Institute Pasteur, which is more of a bulk PBMC based model. They used even higher cell density to begin with. So depending on the responses they were generating, they had to tune the cell density as well.
Great, thanks. We have a question for everyone. How will you deal with donor-donor mismatch in your system? So I'll just go back that same order I went before. James, if you want to start.
I'm sure. We work mostly with innate immune cells. So we have a very good mimic for monocytes, THP1 cells. And so that way we avoid the donor-to-donor variation that you invariably have. Because the other benefit of the THP1 cells is generally when you get donor cells, they're activated to some degree, and that's really the variation you get from donor to donor. Whereas with the THP.
Great, thanks, James. Lani.
So I think the sourcing there is difficulty. So for some of the models with example macrophages and good organoids, we did co-cultures donor matched and non-donor matched, and then looked at the difference. For the innate immune cells, it's quite limited, and we just continue with not donor matched. With T cells, we try to do some HLA matching, but it's definitely for long-term culture, so more than 72 hours, it's an issue.
Julia.
Yes, so on our side, as I also mentioned before, depending on the application, we use either HLA matched donors or mismatched donors. We never had yet the opportunity to have exactly matched donors. But I think it really depends a bit on the application and what is important. So what we can see, as I also answered in one of the questions for me, is that we are still able in a short window, like Lenny said, like after 72 hours might become a problem. So normally we treat 2-3 days. So we are still in this window, and we still manage to have isolate the specific effects of the compounds we test in this window, even with non-matched donors. So there might be some effects coming from that, but if you have the right controls, what we observe in our experience is that you can still see something meaningful. Yeah, and then as also mentioned before, I think it's for certain applications where it's really important and it's more specific, the approach is more specific also, then of course it's possible to then look for more matched donors in the HLA typing or directly from the same donor, and then avoid even the small mismatching effects.
Great. Lewis.
Sorry, muted. Yeah, so our models mainly 95%, 99% of the time are just innate immune competent only, so only containing the alveolar macrophage, which kind of limits the necessity to have the donor-to-donor matching. But we have, in some instances, when we've been using donors from primary cells, if our clients want to use primary models, then we have looked at donor-matched cells, and especially when we've looked to incorporate T cells into that system as well. Usually most of the assays we are doing are in the acute time period. So like Julia mentioned, within like the 48 hours is typically kind of the acute endpoint that we'd be working with for most clients. So again, most of these issues don't seem to be arising, especially just in an innate immune environment.
Great. Gautam.
Yeah, so in the model which I presented today, the lymphoid follicle work, there is absolutely no way we can use donor mismatched cells. It's an immune cell model, so you have to use all the cell types from the same donor. But the other one, which I gave a teaser on, immune cell recruitment for the IBD application, the co-culturing with immune cells is not too long to, the panel is, we all are on the same page pretty much to see any effects due to donor mismatching.
Thanks. And Sylvia?
Yeah, similar to the other speakers, it really depends on the duration of the experiment. We have both used donor-donor match and donor mismatch, and it really depends on the duration, since we can even grow tissue samples for weeks and months. In this case, we typically use stabilized cells, and while if the immunotumor crosstalk is more related in short-term investigations, we use autologous cells from same donors.
Great, thank you. We have 2 more questions for everyone, so maybe I'll just ask them at the same time, and then we can kind of go through. But the first question is, what are the membrane types, compatibilities, and MWCO used to emulate the true capillary permeability for different tissues? And then someone else asked, can you culture biopsies, and if yes, what dimensions can they have? So I'm not sure if there's anyone that wants to start with those questions. Or we can go the same order as prior. I'm not sure if James is still on the line here. I can't see him any longer. Maybe we can start with Lani for those 2.
So for biopsies, we can only culture them in our organoplate graft, and then they should be smaller than 1 millimeter, otherwise they don't fit in. And I didn't get the first question.
No problem. I can repeat. What are the membrane types, compatibilities, and MWCO used to emulate the true capillary permeability for different tissues?
So we don't use membranes. We just due to the red ridges in our platform, we can frame ECM and cells directly next to each other. So if we measure a barrier with tear, it's a real barrier of cells only, and we don't get values for an hydrogel only one.
Okay, thanks. Julia?
Yes, so regarding the biopsies, this we never tried on our chip system, so yeah, I think I will not comment more on that. And regarding the membranes, well, we have our own special membrane that we develop in-house, and on top of this, we put ECM coatings. Depending on the model, we can also vary this, and also similar to Lenny, when we then measure a barrier function, is really coming from the cells, because the membrane that we have is very, very thin. It has big pores and so on. It's really just a tiny mechanical support, but the ECM and the cells per se are the ones that then build the barrier. And yeah, so I don't know if this answered the question. I also struggle a little bit to understand it.
Yeah, thanks, Julia. We've also had a clarification, MWCO, molecular weight cutoff values. Lewis, I'll go to you next.
Yes, we have worked with biopsies before. Off the top of my head, the specific sizes, I am not too sure. I'd have to look back through some of the notes. So if anyone wants to contact me directly if they were wanting to look at it kind of in our systems, yeah, drop me an email. And yeah, I can hopefully follow up, because otherwise I'd just be trying to wing it with a value picked out of my head. And with regard to the membrane, no, we're not. We don't necessarily use membranes. We've got ourselves fixed in a transwell setup, and yeah, like but if we were to have the epithelial cells in there, we would actually be measuring a real barrier based on how they're cultured in that transwell setup.
Thanks, Lewis.
Thanks.
And then Sylvia, I'll end off that question with you.
Yes, okay, regarding the biopsy, we have worked with different dimension of patient biopsies coming from different cancer or tissue models. So both the thickness and the overall dimension can be very different, up to more than 1 centimeter size. So this is a benefit because we can really work with the patient pieces of biopsy without any further manipulation, so keeping the overall complexity of the tissue itself. And regarding the membrane, the platform is compatible with the commercially available insert. Therefore, we can work the inserts having different pore size according to the application. When we have to study immunocells activation, we use the 8 micrometer pore size. In other cases, we use even lower pore size. And as I told you, we can also cellularize the membrane and control the barrier function using tier sensors which are compatible with the platform.
Great, thanks.
Maybe
Sylvia.
I can add something because now I
Yeah,
understood a bit
course.
more the question. So it's about
Absolutely.
size. So I can say that for sure, this immune cells are able to transmigrate to our membrane. So I don't have like this cutoff value, but I can say this. And also we've done some permeability studies with up to 70 kilodalton, and this is also able to pass through easily. But again, as I said before, the cell, if you have a tight layer of cells, that's what is blocking the membrane.
Awesome.
That's it. Thanks.
Thanks, Julia. Yeah, and feel free if anyone has anything to add to just turn on your mic and interrupt me. We have another question for Gautam here. Did you consider using mature dendritic cells in the model? Also, did you consider perfusing the dendritic cells through the system rather than seeding directly into the gel?
Great question. The model right now is monocyte-derived dendritic cell model. We have not tried directly getting dendritic cells and then adding to the chip. The other part of the question is the perfusion of dendritic cells within the system. You can do that. Again, I think for the application, we are looking at generating the antibody responses. I'm not sure what incentive it will bring to flow the dendritic cells in this particular model, but you can choose to do that. The system gives you the ability to do it.
Great, awesome. And it looks like we have one more question, so maybe I'll use that as our last one. So it says, people working with modeling of vasculature in their immune competent systems, how important do you think it is to consider the subphenotype of endothelial cells present in organ-specific niches? Not sure who wants to start with that, depending on who's it's relevant for.
I can start.
Thanks, Lenny.
It depends on your question, but if you want to look at specific organs, I definitely think that you should include the primary cells from the organ. So if you work with liver or gut, we often also use primary cells from the liver or the gut and not the general UVEC ones. The issue with the more primary cells that there is more donor to donor variation. So it's giving more problems to have a stable model over time. So it's also a bit dependent how often you want to do and how repeatable it is. And you often need to test at least 3 or 4 different donors to select one.
Great, thanks, Lenny. Anyone else want to input on that question?
I can comment on what emulate models are based on. So our models are based on using endothelial cells from that particular organ. So if our liver chip is liver sinusoidal endothelial cells, so that's what we are aiming for. And that's pretty much similar to what we do for all the relevant models. You can use generic endothelial cells as well, but to aim for better modeling, I think it is better to use the specific endothelial cells.
Great, thanks, Gautam.
Yes, I may add also that, yeah, we also use different type of endothelial cells depending on the application. And again, these endothelial cells have been either directly seeded in the inserts, and we apply directly the fluid flow, which also foster and accelerate the maturation of the layer. In some cases, we introduce protein functionalization with either matrigel or collagen or other type of matrix in order to create a sort of physical barrier, depending on the application.
Great, thanks, Sylvia. There is an additional question that's been asked for all. How often do you change the tissue culture medium, and how do you adjust or complement or change for different tissues, especially if you have a combination of tissues? So maybe I'll go back to Julia and then Lewis, just because you both didn't speak the other question, and then we can continue the rotation.
Sorry, could I ask you to repeat the question, please?
Absolutely, yeah. How often do you change the tissue culture medium, and how do you adjust or complement or change it for different tissues, especially if you have a combination of tissues?
Yeah, so actually in our system, we change them. It depends always on the cell type, but more or less for all the organs we tried so far, we change the medium every 2-3 days. So we can also avoid the weekend. And for which media we use, also for co-culture and so on, for every model we optimize our media so that we have all the important growth factors for the different cell types that we have included, and then we test maybe different combinations till we find the best one. Yeah, so that's a bit how we develop.
Thanks, Julia. Lewis.
Yeah, again, we would probably typically change the media every probably 4 to 5 days. I know with alveolar macrophages, they're actually happier in a slightly more acidic environment. And we have, yeah, again, we've optimized the media over the years that we've been working with them, and the time points that we would typically use to change it. Again, growth factors are really important, but also metabolite concentrations as well. If you're putting 25 millimolar glucose in there as well, then you're going to probably shift the metabolic profile of your cells as well. And is that physiologically relevant? So I guess it's not just the concentration of maybe like your FBS, so if you're working with that, or also growth factors and cytokines. But yeah, I think the glutamine, glucose concentration is really important to pay attention to too.
Thanks, Lewis. Lenny, maybe I can return to you if you had any additions for that question.
Yes, I think our setup is a little bit different due to the hydrogel. We have a kind of barrier. We see that growth factors go to the other sides, but it's really slow. So in 48 hours, I would say around 1% of, for example, a chemokine reaches the other side. So we can culture with 2 different medium and 2 different sides. At the moment that it's more vascularized, it's getting a little bit more complex, and then we do testing which growth factors and medium components we need to sustain both tissues.
Thanks, Lenny. Gautam.
Yeah, so I agree with the panel here, and also I don't think there is one size fit all answer here. For Emulate platform specifically, we have a reservoir for the media which can hold up to 4 ml of media. So depending on what flow rate you are using, you will be changing the media accordingly. But also it needs to take into other factors in consideration, like stability of the cytokines, how often you need to replace them. So it depends on what model you are generating, how you are generating it, and what flow rates you are using for that.
Thanks. And Sylvia.
Yeah, generally we have seen that we can change media just once per week because due to the fluid flow, which improves the diffusion of drugs but also of nutrients, one per week it's enough. We have also checked with specific sensors the oxygenation and the proper amount of nutrients into the chambers. And when we have a different culture, so a co-culture of different type of cells or tissue, we have seen that we can also use the different media in the two compartments, the bloodstream and the tissue compartment, meaning a different cocktail of growth factors or different media, so DMEM and RPMI or whatever. And this gradient is maintained at least for 1-2 days. Of course, the diffusion and the gradient decrease according also to the tight junction and to the barrier function of the layer of cells which separate the two compartments. But yeah, it's nice that we can use different media in the two compartments.
Thanks, Sylvia. And I guess just a bit of a follow-up on that question, if anyone can comment. Someone says, by changing the medium, you're not emulating the true tissue/organ milia. It becomes artificial as the cytokines and lymphokines are removed and have to be built up and accumulated. So if anyone wants to address or comment on that, I guess more of a comment than a question.
Do you mind repeating it, Lauren? Sorry, I missed it.
Yeah. So it says, by changing the medium, you're not emulating the true tissue/organ milia. It all becomes artificial as the cytokines or lymphokines are removed and have to be built up and accumulated. And then just asked to have individuals' comments on that.
Yeah, I mean, I can make the comment on the model which I presented today, the lymphoid follicle model. The model is based on to address the same exact question. So the cytokines, to maintain the cytokine milieu, every day the media was recirculated. It was put back from the outlet of the pod to the inlet so it can interact with the serum cells there. But for the other models, that's not the case. We do use a unidirectional flow and we replenish the media when it's needed. But yeah, just a comment there.
Yeah, thanks, Gautam. Any other thoughts or comments on that topic?
I can also add that in our system, we typically use a closed circulation, like what happened in our body, although in the organ on chips it's difficult to recapitulate the clearance and therefore the real comparison is a bit complicated. In some cases, we use also an open circuit, not a closed circuit. So in that case, you can continue to have a circulation of fresh media, introducing reservoirs. So according to the specific needs and the questions, biological questions, in our case, we try to modulate a bit the platform thanks to the flexibility to have the most effective comparison with the in vivo situation.
Thanks, Sylvia. Any last comments on that topic? We're getting close to 30 minutes for our roundtable, so I'll start to maybe wrap things up a bit, but just want to give everyone a chance to comment on that if you would like. And I'll also say, just a reminder, I will be sending out a follow-up email after the conclusion of this by the end of the week to everyone who attended. with a link to recording, as this has been recorded. So we're all interested in viewing the presentations if there's anything you missed, as well as contact information for each of our speakers that we have here today. So if there's any follow-up questions based on what was discussed today, any questions that I wasn't able to get to, then you can feel free to reach out to them specifically.